for having us all here talking about biological complex systems and modeling of biological complex systems. So we start with the next session with Dr. Chetan Gadgil. Without much ado, I would rather get started off by saying that Chetan, I know him for a while, and I know about his work which he does in mathematical modeling of biological systems. So Chetan, would you please get started? You have 30 minutes.
Thanks, and let me start by thanking Arijit for inviting me and his team of students and him for this wonderful hospitality. So I'm going to talk about modeling in a wide variety of contexts, and this is what roughly I'll talk about. I'll try to give you my view on the state of theory and modeling in biology, and then some specific examples. Okay, so I'm going to give you basically a economic justification for what I think or why I think modeling is at the state at which it is at. And to do that, I will contrast it with projects in other disciplines. So I'm going to start with my discipline, which is chemical engineering. So a typical large project is a refinery in chemical engineering, okay? And this is the Reliance refinery, but you can take any refinery. It is a city in itself. So building a refinery is more or less like building a city. There is huge amounts, so this is million metric tons per annum. So the amounts involved are huge, both in terms of money, in terms of mass, in terms of energy. But many or most or all of them are quantified. They are quantified by chemical reaction rates, they are quantified by heats of reaction, they are quantified by pumping requirements and so on. So it is of the order of single digit billion of dollars. The time frame is about 5 years, from starting to build to a working refinery. But it is I wouldn't say a routine thing, but there is almost guaranteed success in the sense that if you come up with the money, the design is not the limiting factor, right? It is, then it executes and it may or may not generate a profit, but that's another story. Okay, another kind of large project in the, let's say, physical sciences or literally rocket science is something that was in the news recently, right? So this aircraft or spacecraft intercepted a comet, Bennu, a small comet, traveling, of course, at a very large velocity, made a soft landing on that comet, extracted a sample, flew away, and returned to Earth at a defined date. Right? So it was very precise, deterministically calculated maneuvers that have been successfully executed. The cost is in the same ballpark as the chemical engineering project. Right? Of course, there is also a reasonably high probability of success of such ventures as well, although maybe not as high as a refinery project. In contrast, a large project in biology is a drug development project, right? And this has the same kind of time scales that we are talking about. There's the same kind of funding that we are talking about. So this is development of a drug after the target is identified, after the molecule is identified. Okay, it is, we're not even talking about the science that goes behind that process. It's just pure development. But the success rate is abysmal. Okay, so it is, you can imagine if even the chairman of ISRO goes and says that, well, give me about $1 billion, but there's about 1 in 20 chance that I will actually end up with a successful mission. That is not going to work, and it is also correct. So there is this dramatic difference between biology and the ability, our ability to execute large, ambitious programs in biology and our ability to execute large, ambitious programs in other fields. And my explanation for that is theory. okay, that there is no theory, quantitative, generalizable in biology yet. For instance, we use principles of thermodynamics, kinetics. In physical sciences, of course, there's electricity, magnetism. They all have laws, quantitative laws that relate, that enable design. But we do not have such, we are not at that stage in biology yet. Right? We are at the stage where even this year, right, papers in very good journals have expressions like, in our hands, we get this result. Okay? Which means that the reproducibility is not very high. And this is not to fault the people doing the experiments. This is the state of the field. Okay? There is so much unknown that it leads to, in our hands, level of results. There are no real models that work across scales in biology. And to add to it, biology itself is far more complicated than, I would say, rocket science even. Because of what, as the previous speakers have pointed out, a large degree of loops, positive and negative feedbacks that lead to paradoxical and results in the literature that lead to observations that are not necessarily intuitive. Right? So then what is, to me this is good. I mean, in the sense that it is a good time to be working in this field, in the field of theory in biology, because this is the 1700s of physics. Okay, so there is lots and lots to be explored, and there is lots and lots to be discovered. However, practically speaking, I like this quote, where especially in biology, the point of a model is not a grand unified model that explains end-to-end anything. So a model is a specific prediction. So we want to answer a specific question. It's only about certain aspects of the world. It's only about a subset of this system. I don't know where this pointer is going, but I'm not handling it. So, and the other point is to organize experimental observations, okay? Which means to organize observations from disparate labs into one conceptual or theoretical framework. And always remembering that, as in all fields, models are wrong, but some are useful. So what do we do? Right? So the way we start formulating models is by making choices. And the choices could be whether to treat our system as individual entities, whether to count molecules, cells, etc., or to treat them as a continuous whole. I have no idea where this is coming from, okay? So if it wanders, there's something stochastic about it. But anyway, so we have a choice between whether it should be deterministic or continuous, or discrete or continuous. We have a choice between the system, we should treat it as deterministic or have an element of probability in it. And we have a choice of whether we want to treat cells, for example, as a homogeneous test tube, or we want to treat individual entities within cells, we want to account for diffusion within a cell. Right? So whether we want a mixed system or a spatial system. All examples I will talk about are these. So they will be continuous systems that are well mixed and deterministic. Right? So I will describe them by ordinary differential equations. So 4 examples. The first is a seemingly unintuitive observation about GPCR activation. Okay? So the experiment is as follows. You have a GPCR that is treated with its ligand, right? So this is a receptor, it has an extracellular ligand to it, and ligand is always present for the purpose of this experiment. And the extent of activation of this receptor depends on the G protein that binds to this ligand-bound receptor. Okay, so it's a ternary complex. The G protein itself is of two types. One is the cognate G protein, the native one, and the other is a non-cognate one. So obviously, if the cognate binds, activation is larger. You get a larger extent of activation. If the non-cognate G protein binds, you get a lower extent of activation. However, the interesting observation was that if you pre-treat this G protein with the non-cognate and then add the cognate, you get a higher expression than if you just have the cognate. Okay, so it seems that something which is not supposed to really bind, or not supposed to bind very well, is enhancing the effect of the other molecule that does bind well. Okay, so why is this happening? And of course, I'm going to show you that our simulations captured this phenomena, but the state of the art in G protein or any really receptor ligand binding and activation are such models, cubical models, where the protein, the receptor exists in different states: bound, unbound, activated, unactivated, and so on. Okay. However, all the analysis, because ligand receptor binding and transitions are typically reasonably fast compared to cell division times, all analysis are pretty much steady state analysis. Okay. And also they are limited to one kind of G protein. So there's no competition that has been considered before. What we did was we built slightly more complicated model. This is like a cube in a cube kind of structure. This is actually a prism in a prism. So it's called a tesseract. And this was one of the points of intersection between my teenage kid and me. Okay, because somehow kids of a certain age, and maybe you guys too, I did not know what a tesseract was, for instance. But a tesseract in current, I don't know if anybody has any idea of where you have seen this word, that a tesseract apparently to kids of a certain age is able to contain an infinity stone. Okay, so, but that apart, in the GPCR world, we have this geometric structure that represents transitions between G protein receptor states, whether bound, unbound, active, inactive, etc. However, what we were able to show that even for this complicated model, we can analytically carry out basically pen paper calculations and show that such kind of priming that is seen is not possible. It is not possible independent of the concentrations or the binding affinities or any parameter. So it is independent of any parameter. Yeah, such things are just not possible at steady state. We then went on to show that this is actually a transient effect, which was something new in this field. Okay, and that is where I'm going to stop this story, interesting as it is, and move on to the next story, and hopefully allow time at the end for discussions. Okay, so the next so this was in the test tube, really. Right? So next we move to cells in test tubes, right? Which is this system of mammalian cells which take up nutrients, and I mean, the collaborator I'm working with is interested in using them to make proteins of therapeutic interest, such as monoclonal antibodies. Now, this productivity of the product that these cells produce is influenced by the composition not only of the carbon source, but also of the amino acids in the medium. So it is of interest to know that if I change the concentration of amino acid, what will happen to the cells, or at least how much will go inside the cell, right? And this simple looking analysis is complicated by the fact that it is a very combinatorially complex system. So there are obviously different amino acids that we are aware of, but there are also multiple transporters that carry these amino acids from outside to inside, and in many cases also from inside to outside. These transporters themselves are broadly of three types: uniporters, which are unidirectional; symporters, which require a co-substrate to be transported; and antiporters, which transport one substrate in one direction and another in the other direction. Now, there are different numbers of uniporters, symporters, antiporters. All of the amino acids, to some extent or the other, interact or are able to be transported by all of these transporters. So, therefore, it is difficult to intuitively think of what would be the effect of changing one or multiple amino acids or transporter levels. So what we did was went ahead and build a model specific to CHO based on expression data of all these transporters and the amino acids with known binding affinities and transport rates. Okay. So this was a model. We did some simplification to get an analytical expression for the flux of any particular amino acid at given constant amino acid external concentrations. Okay, that was a simplification. However, as you can imagine, this is able to actually dissect the contributions of different transporters and competing amino acids. So the proof of the pudding is comparing the model predictions to data that the model has not seen, which means data that has not been used in construction of the model or deciding what the parameters of the model are, and it does an okay job, given that it is a completely different system, different lab, different everything that is where this data comes from, right? So again, it is not an exhaustive model. Clearly, amino acids do get metabolized. Clearly, there is regulation of transporter levels and so on, which we have completely ignored. But the question is it useful? So for this limited purpose of trying to dissect contribution of transporters, trying to understand the effect of interactions between amino acids, it is useful. So moving to the third example that I have, this is from such cells to growth in reactors, and in particular fermentation for the production of these human milk oligosaccharides that are substitutes for, they are used in infant baby formula, and there's a big market for them. But we will not get into that. So one particular such oligosaccharide is this 2-fucosyl lactose. And you can think of it as lactose and fucose, two monomers, let us say. They are brought together by an enzymatic process that goes on within the cell to form this 2FL, right? So you need some source of lactose and some source of fucose. Now, of course, the beauty of metabolism is that, and of course you need some enzyme, right? So you need a lot of cells will make a lot of product. So you need to supply these two precursors, and you need something that will make the cells grow. Right? And the good thing about metabolism is that if you just give glycerol, ideally some amount of fucose and some amount of lactose will form inside the cell. But it will not be enough to generate the kind of product that you want. So ideally what you want to do is externally supply these precursors. Supply also something, a cheap substrate that will make the cells grow, and hope that this whole system results in the formation of product. Now, of course, this fucose will also go and do help in growth. So you want to block off these pathways that make the precursors do things other than contribute to product. So there's a lot of strain engineering that is done. And these are all the various strains that have been generated. I don't expect you to know any of them. We have analyzed 3 of these strains and basically built a single model that captures that whole metabolism that goes on inside the cell, together with growth kinetics and product formation kinetics that is non-standard. So one substrate leading to one product is a standard monod kinetics and other models. So what we were able to show is that the model predictions, which is all the solid and dashed lines, are very good match really with the experimental data, which is all the symbols. And this very good match, of course, is not here. The calibration and validation are not different sets. Okay, so it is a good match, not in terms of its predictive power, but its power to capture this system. So for instance, if instead of starting the feed here, if you start here, the model will be able to capture the kinetics of growth and product formation. So this is a good model for the limited purpose of process optimization. Right? It is not a good model to to predict multiple changes in the metabolism. Right? So again, this again goes on, right? So there are 3 different kinds of mutants, so 3 different growth curves, etc., all of which can be captured. It is also captured, this whole model structure can also be used for other oligosaccharides. And so this is another oligosaccharide, and I won't really bother you with the details of that. Okay, so in the last part, I will move on to a very different system, which is tuberculosis. And tuberculosis is a very not just complicated but also complex system. And I'm sure Dheeraj will talk a lot about it, so I will skip this whole slide, okay, in the interest of time. There is great complexity in both the kinds of bugs, where they are located, which drug does what, etc. Okay, but what I will focus on is not pulmonary tuberculosis, which is that of the lungs, but extrapulmonary tuberculosis, which is a large fraction of all TB cases. Of course, in India, that number is huge. I mean, these are COVID level numbers, and it is not very well investigated. So this is what we overall want to do, is to have a combined model that looks at how the bacteria grows, mutates, resistors, persisters, etc., in the presence of an antibiotic, and including the effect of immune system components. So to this end, what I will present now is just a model which is new in the sense that it includes compartments that are associated with extrapulmonary TB that were not present in the literature. Okay? Now this kind of model is called a physiology-based pharmacokinetic model, where the whole body is thought of as vessels, or reactors or compartments. Each compartment is well mixed, and into each compartment flows blood in and out. Drug flows with the blood, partitions into the compartments, and the remaining flows out. Right? So it is like saying that I have a system of tanks with a tracer added to it, which is the drug, and what is the concentration of that ink in each tank. Right? So this is the physical system. There are a lot of parameterization that is available. So equations are not very difficult to frame. And we follow the same process of we estimate parameters. The parameters that we don't know, we use experimental data to estimate. Then we test the model to see whether it works with data that has not been used in the calibration. And if that happens, then there is some confidence in the predictions that the model makes. Right? So this is calibration. So this is data that is been published, and on the 4 frontline TB drugs. These are part of the standard cocktail of drugs that is prescribed if you are diagnosed with TB. And these are basically time versus concentration data. And the model does a good job of capturing this data, which is not unexpected, because this is the data that is used for calibration. Right? So some of the parameters that are not known or available in the literature are estimated using this data. Right? But here the only thing that is changed is the drug dose that corresponds to that particular experiment. And even then, the model seems to do a reasonable job of capturing the drug kinetics. So we have some confidence in the model, and we can now think of the completely different compartments which have neither been used in calibration or validation, and how does the model predict. So this is some data available for a particular compartment. You can see that for some the match is terrible, but for others it is okay, given that. So as you will see in all these results, right? In the first result, it is very tight. It's a very tightly controlled system in vitro. So the accuracy is good. The confidence in the data is good. This is in vivo data. So the natural spread in the data is also good. So the expectation from the model varies accordingly. What the model can do now is predict at the EPTB sites if the antibiotic concentrations are lower than the values that are required, the minimum inhibitory concentrations, the critical concentrations, or they are higher. And if they are higher, what fraction of the time are they higher? Okay, so this is a tool to help thinking that if the same dose is given as is prescribed for pulmonary TB, will it work effectively for extrapulmonary TB? Right? So all these, I hope I have shown, are specific models designed to answer specific questions about small systems. And what I have not shown is many other things that are going on in the lab. Some theory of how motifs react to learning kind of impulses, and other experiments that we are trying to analyze, including purely chemical systems. So in short, if you are a student or you know a student interested in either PhD or like Rahul, I'm going to make a sales pitch. So if you have, we do have positions available. Okay, so I'll stop here. Thank you all. All the persons in italics are the students who have worked on it, so they deserve the applause. But thank you.
Thank you, Chetan. That was a wonderful exposition for both what is not possible with models and what is probably possible with models. So with that, we have a few minutes for questions. So are there any questions? Any questions? Yeah.
That was really nice, Chetan. Great overview from your colleague.
Yeah.
My question is related to your second problem, and I was really interested in understanding the failure modes of that model. When you see for alanine, for aspartate, etc., you're not able to predict the uptake using your model.
Yeah.
Initially, I thought it was because maybe the uptake rates are very small, like for aspartate, but even for alanine, where there's like a huge rate.
So there is an explanation for that, which is metabolism, I think. I should know this, but I will go back and look it up. This is all published work. That was also one of the things that I did not point out, that even if I have given this hand-waving view, there is a detailed exposition of all the models, including the fact that it doesn't work. Especially for alanine, I think we are not capturing the fact that it is both ways transport, as well as it should. But you're right. There are two, I think, or three, in fact, amino acids where it is qualitatively different. The model predicts uptake, whereas it is, the data suggests that it is coming out of the cell. So those we are fully analyzed, but I do not recall off the top of my head. So sorry.
Maybe we can discuss in
Yes,
Pune.
absolutely. Yeah.
Chetan,
All right.
Yeah.
Any other questions? Yeah.
great to see the length and breadth of topics covered in your group.
Thanks.
My, of course, question is regarding the last part, the
Yeah.
PTB. So I know you shared the paper, but I'm guilty of not reading it entirely. So the tissue partitioning from, from blood,
Yeah.
for every molecule will be different, right?
Yes.
Depending on its own partitioning coefficient.
Yes.
So how did you get that parameter? I mean, that's the most fundamental
Yeah,
to
absolutely.
see the distribution.
So there is this data, so it is in effect a correlation based on the chemical properties of the drug. So for example, the log P values and so on that are experimentally known. So log P is oil water partitioning coefficient, okay, roughly speaking. So those are measured routinely for all drugs. And there are these correlations that take those values coupled with the chemical structure of the, what is the electronegativity, what is the size, and they relate it to tissue partitioning. So this Rogers and Rowland method is what we have used, but there are 2 or 3 available. They all come up with similar numbers. So
because
these are purely,
we are actually experimentally doing the same thing.
Oh, great. Well, then we
To
should
look at
definitely
different
talk
tissues, all 14 drugs, how much of these drugs are going at different tissues.
Okay, we definitely have to talk then.
It will be very excellent to
No, this is not our work. This is already published, and it is not specific to TB drugs either. So it is in general, how do chemical entities distribute based on their in vitro measurements of size, as I said, partitioning primarily, oil water partitioning and things.
Thanks.
Okay, maybe we have scope for one more question, and after that, we'll have to take it later because we'll be running out of time. Sorry for that. You can take it up later if that is all right. Yeah.
Sir, I have a question regarding the problem number 1, in which the non-specific protein binds to the ligand, which increases the binding affinity of the cognate protein molecule. So is it necessary every time that this interaction has to be increased? So there might be benefit without increasing the interaction also.
Right. So it is not, and it is a time dependent thing. So as I said, if you go all the way to equilibrium, it will not affect it actually, because it is then just a thermodynamic. In fact, it will reduce it because there's a finite amount of G protein available. And now there are 2 things competing for it. Maybe one has very low affinity, but it is still has to go down. It cannot go up, right? So at very large times, it is clearly the opposite effect. With time, we have shown how the effect, the combination of binding, relative binding efficacy and concentration and time. So depending on where you measure, depending on how much you add, the results qualitatively change. But it is captured in the model.
Okay, thank you.
Perfect. I think we will have to wait and take the questions later. So let's give a round of applause to Dr. Chetan Gadgil for that wonderful exposition.
Thank you.